Hello everybody, this is our report for today and we are talking about bacteriophage, and a few questions might be answered, which is already here in my phone. So today, let's begin. Bacteriophage, or also called phage or bacteriovirus, is a certain type of virus which infects a bacteria. The certain virus will go to the host, which the bacteria is the host, and insert one of its genes in order for it to reproduce. Another example would be a hornet latching its eggs inside a host caterpillar in order for the eggs to hatch and eat the remains inside of the caterpillar and then grow to become a full adult. Bacteriophages were discovered independently by Frederick W. Thorne in Great Britain in 1915 and Felix D. Harold PewDiePie. In 1917, Dr. Harold coined the term bacteriophage, meaning bacteria eater, to describe the agent's bacterial ability, which was seen in the microscope. Bacteriophages also infect the single-celled prokaryotic organisms known as archaea. The characteristics of bacteriophages is that thousands of varieties of phages exist, each of which may infect only one type or a few types of bacteria or archaea. Phages are classified in a number of virus families. Some examples include Inoviridae, Microviridae, and Rudiviridae, and also Tectoviridae. Like all viruses, phages are simple organisms that consist of core genetic material, or nucleic acid surrounded by a protein capsid. The nucleic acid may be either DNA or RNA, and may be double-stranded or single-stranded. There are three basic structure forms of phage: an icosahedral, which is 20-sided, head with a tail, and a icosahedral head with a tail and a filamentous form. So basically, you can search that up in Google, but later this will be edited. Next, we have the life cycles of a bacteriophage. During infection, a phage attaches to a bacterium and inserts its genetic material into the cell. So basically, it inserts its genetic material inside that prokaryotic cell like the archaea in order for it to multiply or reproduce. After that, a phage usually follows one of two life cycles: lytic, which is virulent, or lysogenic, temperate. Lytic phages take over the machinery of the cell to make phage components. They then destroy or lyse the cell, releasing new phage particles. So basically, after injecting the genetic material inside the cell, that certain genetic material will grow to become a new bacteriophage and burst out of the cell, eating what's left of its dead body or the remains of it. Lysogenic phages incorporate their nucleic acid into the chromosome of the host cell and replicate it as a unit without destroying the cell. Under certain conditions, lysogenic phages can be induced to follow a lytic cell. So basically, the lysogenic is where they insert their genetic material to the chromosome of the cell, but it does, instead of bursting out or destroying the cell, it won't destroy the cell, but then it may also follow the lytic cell cycle again. So basically, there's a chance it may burst out of the cell. Other life cycles, including pseudolysogeny and chronic infection, also exist. In pseudolysogeny, a bacteriophage enters the cell but neither co-ops cell replication machinery nor integrates stably into the host genome. Pseudolysogeny occurs when a host cell encounters unfavorable growth conditions and appears to play an important role in phage survival by enabling the preservation of the conditions have become advantageous again. In chronic infection, new phage particles are produced spontaneously over long periods of time but without apparent cell killing. So basically, during pseudolysogeny, it's basically the same thing with lysogenic, but with the same part that they don't kill the cell or cell killing, but it takes a long period of time in order for that genetic material to become a full-grown bacteriophage. Thank you everybody for listening to our group report. I hope you learned something about bacteriophages and their life cycles. Thank you again. Goodbye!